This is the second part of the prep for lecture 28. And I just want to talk a little bit about the proximal distal patterning and the anterior posterior patterning. And so the proximal distal patterning is driven in part by the progress zone, which I already mentioned in the previous lecture, which is a zone immediately under the apical ectodermal ridge, which we always abbreviate this way. And this is the FGF signaling here is controlling kind of the outgrowth, and as the limb grows out, the rapidly dividing stem cells in this region remain beneath the ridge, but they leave behind a set of determined cells that once they quote unquote leave the progress zone, they start the differentiation process. But it's not like they're moving out, it's that the progress zone continues to move with the apical ectodermal ridge. These are rapidly dividing cells, and then the cells that have taken on certain fates, I'm going to form the humerus, I'm going to form the radius, and so on. Those are left behind. They stop moving along with the AER and the progress zone. So that's kind of the proximal distal story. There's also a polarizing region, which is often called the ZPA, the zone of polarizing activity. It's right on the most posterior part, posterior and sort of medial, because or proximal, I guess we'd say, because it's towards the body axis. This is a source of signals for anterior versus posterior patterning, with the signals that have the strongest signal being posterior, and then there is a gradient towards the anterior with less and less. And over time, we know that and I'll show some data on this in a minute, but that the most proximal structures form first, and then the more distal ones as outgrowth takes place. Okay, so this, for some reason, this always reminds me of a nose, and the nostril is the place where the zone of polarizing activity is, and there's a high level of sonic hedgehog expression, and that expression grades out towards the more anterior. If you take the zone of polarizing, suppose you excise it from one embryo and then put it to the anterior part of another limb bud on a different organism. Just think about how you would do this so that you would know which tissue came from where. You can find a limb that has a mirror image duplication, so posterior is going to be like the baby finger, and then there's going to be a baby finger here, and then there's going to be the next finger and the next, and so on. So you'll get this. This is badly drawn because I can't draw, but the idea is you'll have a mirror image duplication with the posterior structures on the outsides of it and the anterior structures on the insides of it, because you now have two sonic hedgehog gradients with the strongest and then the weaker signal, and then the stronger and the weaker signal, and then that gives you this mirror image duplication. And we know that the extra digits that that develop come from the host rather than the transplanted tissue. The transplanted tissue may participate in the formation of one of these digits, but the others that form come from the host. And so why is it important that we know this, and how do you think we know this? The apical ectodermal ridge is important for outgrowth. If you remove it at any point during development, then the outgrowth ceases. So you don't get any further development. If you remove it right after formation at 3 days, which is approximately the earliest time you could actually see the AER, then the wing that develops will have a humerus and maybe some partial radius and ulna bones. So it's only got the most proximal of the structures. If you wait even half a day later, you get almost complete radius and ulna. Another half day and you get complete radius and ulna and some of the carpals, the wrist bones, and so on. And so what this means is that this shows you how these tissues are specified and then determined over time. And that the missing structures are always distal to whatever is left behind. So you're never missing the radius and ulna, but you do have the carpals or something like that. This tells us that the progressive development starts with the humerus, and then the radius and ulna, and then the carpals, and then finally the digits. So that's our timeline. Now, if you remove the apical ectodermal ridge, that usually stops the growth of it and the specification of all the parts of the limb. But you can take beads that are soaked in a number of FGFs, not just the ones that are normally expressed there, but FGF4 or FGF8, even though we know that the signals that come under here are FGF10, and then the AER is supposed to signal back with FGF8, right? So if you replace the apical ectodermal ridge with beads soaked in FGF8, they can take on that function and the signaling back and forth. So the cells immediately surrounding here will start to form a new AER, and will allow for the outgrowth. But it turns out that if you use FGF4, which is not part of this process normally, it will also do that. You can also graft a different AER from a different embryo and from a mouse embryo, and it will just go ahead and integrate and then form a normal limb. And so the apical ectodermal ridge of the mouse and of another chick embryo and so on can just easily substitute, as well as just providing FGF signals, which are responsible for this outgrowth. So there are complications because there's there's 4 FGFs that are expressed in the apical ectodermal ridge in different amounts. And they can substitute for one another. So I mentioned before with TGF-beta work, you can basically induce a response in animal cap cells, in frog embryos, by culturing with beads soaked in different TGF-betas, even if those are not the ones that normally do the job in the frog embryo. And so we have the same situation with these FGFs. And if you're trying to get a phenotype from genetic mutation, you have to knock out 3 of these 4, because you need 2 different ones to signal back and forth. So you need a different one sort of in the cells underlying the apical ectodermal ridge, and then a different signal back. So if you've got even 2, you could get this kind of back and forth, right? So you have to knock out 3 of them, because then one can signal, but then there's no response, and then you lose the whole positive feedback that leads to that, outgrowth and further development. So limbs are pretty important to an organism, and so they're just like with the nervous system development, there is a certain degree of backup. Even FGFs that aren't normally responsible for the full sort of outgrowth and development are able to replace the ones that are, if they are missing or genetically knocked out. And finally, this is what I'm going to finish on for this lecture. The formation and that that outgrowth of the limb bud involves not just cell division, but changes in orientation. So cells migrate from the lateral plate mesoderm towards the developing limb bud, and then the plane of cell division coincides with how the migrations occur. So at the beginning, before the bud forms, the cells are lined up sort of apical basal, parallel to the body axis. So if these are your somites and this is, your neural tube is going to be here, anterior to posterior. And so the cells, they line up, the arrows indicate their orientation. So the apical basal polarity is the same in all of them. And so they, quote unquote, point towards the posterior. And as migration occurs, they're migrating kind of towards the posterior. And the cell divisions allow for that. So if you're trying to move in this direction, it makes sense to divide your cells this way, because now you've made more cells in the row and you're expanding the tissue, right? If you change the orientation of cell division perpendicular to that, then you'd get outgrowth in the other direction. So this is the first part. And then as the bud starts to emerge, and there are the signals, the FGF signals and so on, what happens is the anterior cells from that lateral plate mesoderm, they start moving towards the posterior, but they move along underneath the apical ectodermal ridge. So that's what this part of the division happen, or the migration is happening. So that's how they move. And then the lateral cells orient perpendicular to the body axis, and so they're going to both move and divide that way. So this is another example. We talked about this a little bit in plants, but in this case, altering the plane of division lengthens or widens your tissue depending on how you orient that. So the lateral cells here, they turn and orient towards the bud in a sense, and they start moving in that direction, and that's going to lead to further outgrowth here. And then finally, the bud is starting to elongate. So rather than just a little bit of outgrowth, now we're seeing elongation, and the cells in the center and at the tip. So these are the central cells, and these are the tip cells here, and they're parallel to that proximal distal axis and perpendicular to the anterior posterior body axis. And they are moving towards the apical ectodermal ridge, and all the time that they're moving, there's also division. And so remember that those divisions are occurring as well to make more cells under here. But the dorsal and the ventral cells, they're sort of oriented, polarized towards the ectoderm on the dorsal and ventral sides, and they're they're moving not quite perpendicular to the actual movement of the direction of the outgrowth. So they're kind of broadening the tissue a little bit here. And these first two, these are based on observations and measurements and so on of cell movement in the chick embryo. But this last one were observed in the mouse embryos, and this last one was observed in the chick embryos. And so the information from all three types is put together to form sort of a complete picture. So the main thing to understand is that the cells change their sort of orientation and their migration to change the shape of the tissue, as well as the division that of course causes outgrowth, because you have more cells, so you're pushing the apical ectodermal ridge further out. But there is also this migration and the change in cell orientation, so that you'd have sort of apical basal orientation this way now, and when the cells divide, they divide on this plane to make more cells in this direction. And I think that's all. Yes. So the next lecture will finish the limb formation and talk a little bit more about proximal distal patterning, but we're also going to talk about dorsal ventral patterning, and digit specification, and anterior posterior patterning. And we're not going to have time today. The insect limbs and eyes, they're kind of interesting because they form very differently. Um, than vertebrates, and they're kind of cool, but we don't really have time. Uh, but because we're skipping that, we have time to finish with just looking at experiments that show how limb regeneration occurs and which signals are involved, and that should be, I hope, for students pretty interesting.